So the very last section in this chapter is about leaf structure and diversity. So I'm going to leave it to you guys to learn about leaf diversity, but I'm going to just talk to you really quickly about this figure, because once we get through this figure, then you'll know everything you need to know about leaves. So leaves are where the majority of photosynthesis takes place inside of a plant, and that's why remember, structure meaning function. So their function is to photosynthesize, and so they want to make as best use of all of the gases, the carbon dioxide that they can. So they're really flat because it maximizes their photosynthetic surface, and they're really thin, and that way they're not wasting tissue that's not doing photosynthesis. So here's a cutout of a leaf from a dicot. So we have the upper epidermis and the lower epidermis down here. And remember, here's the trichomes that are just outgrowths of the epidermal cells. They're not actually individual cells themselves. They are just kind of like shoot-offs of the cell wall and of everything from inside the epidermal cell. And then inside here we have all of these parenchyma cells. So they're just ground tissue, just doing jobs, just photosynthesizing the ground tissue. And then in here we have the vascular tissue of the leaves. So we already covered the epidermal cells. Down here, one more thing to notice is the stomata of the epidermal cells. So remember that stomata are the openings in the usually in the bottom of the leaf surface, because that way they lose less gases that way, less transpiration, less water, and they are made up of two specialized epidermal cells called guard cells, and that's what's right here. So remember, when the guard cells fill with water, they open up just a little bit and let the gases in and out, and then when the cell doesn't want to lose gases or lose water anymore, it releases the water from the guard cells, they go limp, and the opening closes. So that's how a stomata works. So that's on the underside, on the epidermal cell down there. Then we have all this ground tissue, which is doing photosynthesis, all these parenchyma cells. There's two kinds of parenchyma cells, and these are called mesophyll cells. And there's two kinds of mesophyll cells. There's the Palisade Parenchyma, Palisade mesophyll up here, and the spongy mesophyll, the spongy parenchyma down here. And so if you'll notice, they're just kind of arranged differently, and you can actually see it in this image down here too. I don't know if my picture is covering it, but this is an actual cross-section of a leaf taken by a microscope. This picture is in your textbook. I don't know what page, but it's in your textbook. So the Palisade parenchyma are stacked very closely down here, up here, and they're doing photosynthesis. And then the spongy mesophyll down here are more loosely packed, and there's a lot of gases being interchanged down there within the spongy mesophyll. But they're just doing photosynthesis. They are taking in carbon dioxide and making sugars. And then they need the water, and they get the water from the xylem, which is this red structure right here. So the xylem is usually on top, and then the phloem's on the bottom in these vascular bundles in the dicot cells. So the leaf is getting their water supply from the xylem cells. It's moving up from the roots, and then they're making all of this sugar, and they're supplying it to the rest of the plant. They're using some for themselves inside the cells, and any extras, they put it into the phloem, and they pump it to the rest of the plant. So water and minerals enter leaf through xylem, and sugars exit leaf through phloem. So they're going from, water goes from roots to shoots, and sugars goes from shoots to roots, the opposite way. And so these are the veins right here of the cell. So if you were to look at of the leaf. If you were to look at a leaf and you could see all of those things branching off, that's actually the vascular tissues right here. So water's being pumped in, sugar's being pumped out. And surrounding the vascular bundles are these things called bundle sheath cells. And so kind of makes sense. This is the vascular bundle, and these cells are forming a sheath around the bundle. So they're the bundle sheath cells. They are just normally parenchyma cells. They're still doing photosynthesis, but their job is to kind of like regulate what's happening inside of the vascular bundle. If you guys will think back to photosynthesis, remember that C4 plants make a modification, and they actually do the Calvin cycle of photosynthesis inside the bundle sheath cell. So that's how they differ. That way they don't have to worry about rubisco fixing carbon dioxide or fixing oxygen inside of carbon dioxide. So they do their photosynthesis inside of the bundle sheath cells. So that's the epidermal tissue up here, the trichomes and the stomata. Then there's the ground tissue, all these parenchyma cells, the Palisade and the spongy parenchyma cells that are doing photosynthesis, making sugar for the plant. And then the vascular tissue, which is supplying water and removing the sugars. And then learn about all the different kinds of leaf diversity. That's very easy, and that's all you need to know about leaves.